Hello, my name is Sanjana Licky, and presenting with me today is Lila Abbas, Fiza Abbas, and Matthew Hunt. Our project is called Minor Changes with Large Implications, Modeling Amino Acid Mutations in SARS-CoV monoclonal antibodies ADR and 362 toward the design of more universal antibodies. We wanted to understand how a more universally effective antibody against a SARS-CoV virus can be predicted using molecular models. SARS-CoV-2 is responsible for the COVID-19 pandemic that continues to have a global impact. The two anti-SARS-CoV monoclonal antibodies, or MABs, ADR and 362, bind to epitopes on the spike protein receptor binding domain in order to neutralize the virus. As seen in Figure 1, the ADR antibody binds to SARS-CoV-1 and the 362 antibody binds to SARS-CoV-2. The space-filled models show the antibodies in yellow and the spike proteins in blue, and the black box shows a detailed view of the 3D interaction that occurs at the binding interface. By comparing these existing antibodies, our research team, as a part of the CREST program with the Center for Biomolecular Modeling, proposed a potentially more universal antibody that could better neutralize future mutations in the SARS spike protein.
Using the existing protein database files of ADR and MAB362, we proposed our new hypothetical antibody, NSU1. We used a protein visualization software, JMOL, so that our collaborators at the CBM could help us print out proposed model. Specifically, after observing trends in the amino acid composition near the binding site of these antibodies, we proposed 4 additional amino acid changes to develop NSU1.
As seen in the results sections, model A through C are various base fill models that show the different binding interactions. So model A shows the ADR antibody bound to the SARS-CoV-1 spike protein. Model B shows the 362 antibody bound to the SARS-CoV-2 spike protein. Model C shows our proposed hypothetical NSU1 antibody that is similar to the 362 antibody with an additional 4 amino acid changes shown in purple. Figure 2 compares how effective the binding of 1-NSU, ADR, and 362 antibodies are to SARS-CoV-1 and SARS-CoV-2. The NSU1 antibody can bind to both coronaviruses and has the highest efficiency in binding to the SARS-CoV-2 in comparison to the existing antibodies. The blue portion of the color-coded table shows the 3 major residue changes between SARS-CoV-1 and SARS-CoV-2. And then the tan portion of the table shows the residue changes between the spike epitopes. The selected amino acid changes for NSU1 tend to be smaller and less polar to make the antibody better able to change as the spike protein may get larger or smaller in future mutations. That is why the changes highlighted in purple in the table and on model C were selected to open up the antibody to a greater degree.
Now for the discussion, NSU1 is a hypothesized antibody that combines the biochemical aspects of the antibodies ADR and MAB362 to allow more space in the binding domains between the antibody and the spike protein. NSU1 is modeled based off of MAB362 with 4 additional mutations that were previously mentioned that tend to decrease the size and the polarity of the antibody. The extra space in the binding domain could allow for antibodies to be even more resistant to future SARS-CoV variations. And these models allow for a deeper understanding of the impact that mutations and antibodies can have on binding interactions with viral proteins.
The applications of 3D molecular modeling include visual representations of molecular processes to enhance student education and enrich hands-on learning experiences. The models may also assist clinical researchers in better understanding the SARS-CoV-2 binding mechanism. We are very thankful that we got to be a part of the CREST program this year through the Center for Biomolecular Modeling, and we would be happy to answer any questions that you may have.